So the topic right now is carbohydrates. Carbohydrates are basically polyhydroxyaldehydes of ketones. We have 3 carbon, 4 carbon, 5 carbon, 6 carbon. The 3 carbon ones are known as trioses, 4 carbon ones are known as tetroses. Then comes pentoses with 5 carbons, and then hexose with 6 carbon. D-glucose is aldehyde sugar, and the combination of alcohol and aldehyde gives rise to a hemiacetal. This is the structure of glucose, which is a 6 carbon sugar, and this is the hemiacetal form. The anomeric carbon, which is the closest to the functional group, the position of hydroxyl group on anomeric carbon gives rise to either the beta form or the alpha form. So if the hydroxyl group is on the right side, it gives rise to alpha form, and if the hydroxyl group is on the left side of the carbon, it give rise to the beta form. So basically, when the position of H and OH varies on C1, basically the anomeric carbon of glucose, या फिर C2 carbon of fructose, they are known as anomers. Then comes epimers. Epimers is basically when the position of H and OH varies on any other carbon. So glucose and galactose are C4 epimers, which means the H and OH group on glucose and galactose are different at the carbon number 4, and uske alawa every structure is same. And then the second example of epimer is glucose and mannose, wherein the carbon number 2 has different position of H and OH. The rest of the structure is same. We can see that the structure of galactose and mannose, and we can compare it with the structure of glucose. So this is the structure of galactose. Again, it is a 6 carbon sugar, 1, 2, 3, 4, 5, 6. And since it is a C4 epimer of glucose, we can observe that this is the 4th carbon, and we can see that the OH group or 4th carbon is on the left side in galactose, whereas the OH group of 4th carbon in glucose is on the right hand side. And again, if we compare the mannose structure with that of glucose, they are C2 epimers, which shows that the OH group of mannose on carbon number 2 is on the left side, whereas the OH group on glucose at carbon number 2 is on the right side. So, and the rest of the structure is same. So these are epimers. Glucose and galactose are C4 epimers. Glucose and mannose are C2 epimers. Fructose is a keto sugar. That means the functional group is a keto group. Again, fructose is also a 6 carbon sugar. This is the hemiacetal form. Ring structures are known as Haworth projections, and the position of the CH2OH and OH with respect to the anomeric carbon give rise to the alpha configuration or the beta conformation. When glucose is present in ring structure, it is known as pyrin ring, and when fructose is present in ring structure, it is called as a furan ring. And this is the structure of glucose, the pyrin ring, and this is the structure of fructose, the furan ring. Moving on to the ring structure of mannose, we can observe over here 6 carbon, 1, 2, 3, 4, 5, 6. And similarly for galactose, we have 1, 2, 3, 4, 5, 6. These are the ring structure of mannose and galactose. If we talk about the stability of conformation of glucose, the stability, the ascending order of stability goes starting from the least stable conformation, that is planar. It is least stable because it has most amount of energy, followed by half chair, which is comparatively more stable to planar, then the boat conformation, then twist boat conformation, and the chair conformation. So in case of chair conformation, it is the most stable structure because it has the least amount of energy. If we want to calculate the number of isomers of a particular structure, the formula for it is 2 raised to the power of number of chiral carbons. So chiral carbon are basically the carbon which have all the 4 functional groups or all the 4 side chains attached to them are different, those carbons are called chiral carbons. All monosaccharides are reducing sugars. Basically, for a sugar to be reducing, the anomeric carbon has to be free. So in case of all monosaccharides, the anomeric carbon is free. Therefore, all monosaccharides are reducing sugar. Talking about disaccharides, first disaccharide is maltose. Maltose is made up of two glucose units. Uh, Two glucose units with the removal of, they combine each other uh, from first and fourth carbon. A water molecule is removed and we get the structure of maltose whose IUPAC name is alpha D glucopyranosyl 1,4 alpha D glucopyranose. Maltose is made up of two glucose units, and if it's made up of glucose and fructose ring, we get alpha D glucopyranosyl 1,2 beta fructofuranosyl. Talking about invert sugar syrup is basically a solution in which the conversion of dextro to levorotatory occurs. So if we have sugar, the specific rotation of sucrose is plus 66.5 degrees, which is basically a dextrorotatory, and we create a solution because sucrose is composed of glucose plus fructose. So glucose has a specific rotation of plus 52.4 and fructose has that of minus 92 degrees. So when it's broken down into glucose and fructose, it gives us the average, the total specific rotation of the solution becomes minus 19.75 degrees, which is levorotatory. So basically, the dextrorotatory syrup converts into levorotatory. Converting of dextrorotatory to levorotatory, we call it as invert sugar syrup. Second disaccharide is lactose. It is composed of combination of glucose and galactose. This is beta galactose combining with beta glucose. Again, the removal of one water molecule. Uh, first carbon of galactose forms a bond with the fourth carbon of glucose. And the resulting structure, we have lactose, which is beta D galactopyranosyl 1,4 beta D glucopyranose. And lactose, as we all know, is a reducing sugar because it has a free anomeric carbon. Then we have trihalose. It's disaccharide made up of two glucose units and it's found in fungi. The two glucose units in trihalose are combined by alpha 1,1 linkage, and trihalose is a non-reducing sugar. Then we have mellibiose, which is found in legumes and it is formed with 1 unit of glucose combined with the unit of galactose through alpha 1,6 linkage. Cellobiose, 2 unit of glucose combined by beta 1,4 linkage. Both mellibiose and cellobiose are reducing sugars. Coming on to polysaccharides, the carbohydrates that are made up of multiple units. So first polysaccharide is a starch. Basically, starch is a polymer of glucose, and plants store glucose in the form of starch. There are two parts of starch. One is amylose and the second one is amylopectin. First, talking about amylose. Amylose is about 15 to 20%. It's a non-branching helical structure. The linkage found in amylose is alpha 1,4 linkage. It's a straight chain structure. Coming on to amylopectin, 80 to 85% is amylopectin. It's a branched chain structure and 24 to 30 glucose residues are linked together in by alpha 1,4 linkage in a straight chain, and at the branch point there is alpha 1,6 linkage. Gibberellin, they are responsible for seed germination because they enhance the alpha amylase activity. So what happens is starch, the action of alpha amylase occurs, it is converted to glucose. When concentration of glucose is high inside the cell, the seed, it creates a hypertonic solution due to which water enters and it leads to the swelling of the seed and ultimately germination of seed occurs. Next polysaccharide is glycogen. So animal store starch in the form of glycogen. It is a very highly branched structure and it is even more branched than a amylopectin. Insulin is another polysaccharide. It is a polymer of fructose, and fructose units are linked together by beta 2,1 linkages. Next polysaccharide is dextrin. They are basically bacterial and yeast polysaccharides, and poly D glucose units are linked via alpha 1,6 linkage, and at branching, they have all of the dextrin molecules have alpha 1,3 branches. A few have alpha 1,2 and alpha 1,4 branching in case of dextrin. Cellulose is the chief constituent of the framework of plants. It's not soluble in any ordinary solvent, so special solvents are required to solubilize cellulose. It consists of beta D glucopyranose units, which are linked by beta 1,4 bond, and they form long straight chains. And cross-linked hydrogen bonding is present in cellulose. Humans cannot digest cellulose because they don't possess the enzyme which digests beta 1,4 bonds. So the enzyme has alpha amylase. It is also called Tyalin. For general information, the receptors for steroids are present in cytosol. The receptor for thyroid are present in nucleus. Next polysaccharide is chitin. It is an important structural polysaccharide of invertebrates, and it's present in exoskeleton of all the crustaceans and insects. And it is a polymer of N-acetyl glucosamine, which is linked by beta 1,6 glycosidic bond, and it's present in fungal cell wall. Talking about mucopolysaccharides, which are also known as glycosaminoglycans, they consist of chains of complex carbohydrates and they are characterized by content of amino sugars and uronic acid. When chains are attached to a protein molecule, it's known as proteoglycan. So basically, glycosaminoglycans, the amino sugars and uronic acids are complex with carbohydrates, and in case of proteoglycans, these are connected with a protein molecule. The chains are connected with a protein molecule. Example of glycosaminoglycans are hyaluronic acid, chondroitin 4 sulfate, and heparin. So hyaluronic acid is basically made up of beta glucuronic acid and N-acetyl glucosamine. Chondroitin 4 sulfate is made up of beta glucuronic acid and N-acetyl galactosamine sulfate. Heparin is made up of sulfated glucosamine and sulfated iduronic acid. Heparin is an anticoagulant. Talking about examples of proteoglycans, we have syndican, aggrecan, and beta-glycans. Now moving on to the metabolism of glucose. So glucose is metabolized through a process known as glycolysis. Glycolysis is basically the splitting of glucose molecule. It takes place in cytosol of cell, and it's the glycolysis part is common for both aerobic and anaerobic respiration. Glycolysis is also known as Edmund-Markov-Parnas pathway, or we also write it as EMP pathway. There are 10 steps involved in glycolysis. 7 are reversible, 3 are irreversible. The 3 irreversible steps are first, third, and 10th. Glucose converts to at the end of glycolysis, glucose is converted into 2 pyruvates, and overall 2 ATP and 2 NADP plus, so NADH plus H2H positive are produced. So to keep the glucose inside the cell, it is first converted to glucose 6-phosphate because the transporters are not capable of transporting glucose 6-phosphate out of the cell. So this begins the glycolysis. Glucose is first phase is the preparatory phase. Glucose is first converted into glucose 6-phosphate by the action of hexokinase and magnesium ion is as the cofactor, and one ATP is used to carry out this conversion. Then glucose 6-phosphate is converted to fructose 6-phosphate in the presence of enzyme hexose isomerase, and this is a reversible step. Fructose 6-phosphate in presence of phosphofructokinase 1 is converted to fructose 1,6-bisphosphate, and one ATP is again used in this step. Then phosphofructokinase 1 is also represented as PFK1. Yeah. Then next, fructose 1,6-bisphosphate in presence of aldolase, it's converted to two. It's broken down into two 3-carbon compounds. One is the dihydroxyacetone phosphate, and the other one is the glyceraldehyde 3-phosphate. The dihydroxyacetone and glyceraldehyde 3-phosphate, they are interchangeable through the enzyme called triose isomerase. From here, the payoff phase starts, wherein ATP is produced. In the preparatory phase, ATP is utilized, and in the payoff phase, ATP is produced. So from glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate is formed. Enzyme involved is glyceraldehyde 3-phosphate dehydrogenase. In this process, one NAD plus is converted to NADH and H positive. 1,3-bisphosphoglycerate is then further converted to 3-phosphoglycerate in the presence of enzyme phosphoglycerate kinase, which is also involved in the substrate level phosphorylation. And in this process, one ATP molecule is converted to ATP. Then 3-phosphoglycerate is converted to 2-phosphoglycerate in the presence of phosphoglutarase mutase enzyme. Then 2-phosphoglycerate in presence of enolase by removal of 1H2O is converted into phosphoenol pyruvate, which further by the action of pyruvate kinase enzyme is converted into pyruvate. This enzyme, pyruvate kinase, is also involved in substrate level phosphorylation. At the end of the step, one ATP is again produced. So why have I written, I have multiplied every step with 2, because the dihydroxyacetone step, the dihydroxyacetone formed is also converted into glyceraldehyde 3-phosphate, and it undergoes the same procedure of the cycle to form one more molecule of pyruvate. So these two carry out, undergo glycolysis to form pyruvate. So this is how two pyruvate molecules are formed. The muscles lack glucose 6-phosphate, and fructose 1,6-bisphosphate is a substrate that is exclusive to glycolysis. Hexokinase requires magnesium as cofactor for its activity, because the true substrate of hexokinase, it's not ATP, but it's magnesium complexed with ATP. And hexokinase is also not present in all organisms. Human genome codes for 4 different hexokinase, 1, 2, 3, 4, and they catalyze the same reaction. If 2 or more enzymes catalyze the same reaction, they are encoded by different genes. We call them isoenzyme. This is the enzyme activity graph for hexokinase. So we can observe the Vmax of hexokinase 1 is greater than the Vmax of hexokinase 4. Hexokinase 4 is also called glucokinase, and it's present in hepatocytes, and so its kinetic and regulatory properties are different. So net reaction of, this is the net reaction of glycolysis. We have the substrates over here, glucose and 2 NAD plus molecules, ADP and inorganic phosphate. They give rise to 2 pyruvate, 2 NADH, H positive, 2 water molecules, and ATP. So the delta G, the 2 reactions are involved in this overall reaction. First reaction is glucose with NAD positive gives rise to pyruvate and NADH and H positive. And second is the conversion of ADP plus inorganic phosphate into ATP and water molecule. So the delta G of first reaction is minus 146 kilojoules per mole, and delta G for the second reaction is 30.5, which is 61 kilojoules per mole. So if we add both the reactions, the overall delta G, the Gibbs free energy for the net Gibbs free energy for this reaction is minus 85 kilojoules per mole, which shows that glycolysis is a spontaneous reaction, because we know that if the Gibbs free energy is negative, the reaction is spontaneous, and if the Gibbs free energy is positive, it is a non-spontaneous spontaneous reaction. So we know that formation of ATP is a non-spontaneous reaction because the Gibbs free energy of formation of ATP is positive. However, the breakdown of ATP is a spontaneous reaction because the Gibbs free energy of the breakdown of ATP is negative. Then there comes Pasteur effect. Pasteur effect is basically the inhibitory effect of oxygen on the rate of glycolysis. How oxygen has an inhibitory effect on the rate of glycolysis, that's known as Pasteur effect. During anaerobic conditions, respiration is faster. Normal blood glucose level is 4.5 to 5 millimoles. There are two types of diabetes. Type 1 is insulin dependent and type 2 is insulin independent. Diabetes mellitus type 1. Talking about type 1 of diabetes mellitus, it is insulin dependent. It is an autoimmune disorder. The beta cells of pancreas are killed by the immune system, and type 1 diabetes mellitus occurs in children. In case of type 2 diabetes mellitus, it is insulin independent. It happens with age. The insulin receptors, they change, and due to which insulin is unable to bind to them. Under normal conditions, insulin receptor is present on the cell. Insulin comes and binds to the receptor. Glut transport opens and glucose enters the cell, thereby decreasing the glucose levels. That's how the blood glucose levels is maintained. Examples of a few autoimmune disorders are Hashimoto thyroiditis, pernicious anemia, myasthenia gravis, multiple sclerosis, insulin-dependent diabetes mellitus, Goodpasture syndrome, Graves' disease, systemic lupus erythematosus, and rheumatoid arthritis. Then HbA1c is basically a test for diabetes. It presents glycosylated hemoglobin, in which three levels of tests are done of diabetes patient, A, B, and C. AMA, we find out the HbA1c value, which is basically glycosylated hemoglobin. B is for blood pressure, and C is for cholesterol. So for diabetes patient, HbA1c test three things: A, B, C. A is the HbA1c, B is blood pressure, and C is cholesterol. Diabetes insipidus occurs due to the deficiency of ADH, antidiuretic hormone, or also which is also known as vasopressin. Then coming on to the fate of pyruvate under anaerobic conditions. What happens to pyruvate when it is in anaerobic condition? So first is the lactic acid fermentation. It can go into the lactic acid fermentation mode, in which pyruvate converts into lactose in the presence of lactose dehydrogenase. In this process, one NADH plus proton H positive is used and NAD positive is released. Next, it can also undergo ethanol fermentation. Pyruvate is first converted to acetaldehyde by removal of one carbon dioxide in the presence of enzyme pyruvate decarboxylase, and this acetaldehyde is converted further to ethanol by the action of alcohol dehydrogenase. Decarboxylation, basically removal of carbon dioxide, is carried out by in the presence of thiamine pyrophosphate as cofactor. TPP, we also call it as TPP, thiamine pyrophosphate. And the carbon in case of carboxylation, where CO2 is added, we need biotin as cofactor. Biotin is vitamin B7 and thiamine pyrophosphate is vitamin B1. If we talk about the fate of pyruvate under aerobic conditions, under aerobic condition, the pyruvate undergoes citric acid cycle. Citric acid cycle is also known as dicarboxylic acid cycle or Krebs cycle or TCA cycle. The TCA cycle happens in the matrix of mitochondria. So if this is the structure of mitochondria, there's this outer membrane, we have the inner membrane, and this is the matrix. So in case of matrix, the TCA or tricarboxylic acid cycle or Krebs cycle or citric acid cycle occurs. So the total yield of one citric acid cycle is 3 NADH plus H positive, 1 FADH2, 1 GTP, 2 carbon dioxide. All the enzymes of citric acid cycle, we'll find them in the matrix of mitochondria. However, except for one enzyme, which is the succinate dehydrogenase enzyme. Succinate dehydrogenase enzyme is present in the inner mitochondrial membrane. So here we have the tricarboxylic acid cycle, TCA cycle, Krebs cycle, citric acid cycle. So the substrates, it starts with pyruvate under aerobic condition, converts into acetyl-CoA. So we have pyruvate dehydrogenase complex over here, which comprises of 3 parts. And the 3 parts of pyruvate dehydrogenase complex are: first is the pyruvate dehydrogenase, second is dihydrolipoyl dehydrogenase, and then dihydrolipoyl transferase. So the conversion of pyruvate to acetyl-CoA involves the pyruvate dehydrogenase complex. From acetyl-CoA, it is converted into citrate, which is a 6-carbon compound. Then in presence of aconitase, it's converted to cis-aconitate, again a 6-carbon compound. In presence of aconitase, cis-aconitate is isocitrate is produced, 6-carbon. Then oxalosuccinate is produced by the action of isocitrate dehydrogenase 6-carbon compound, which further produces alpha ketoglutarate, which is a 5-carbon compound. Then from alpha ketoglutarate, we have succinyl-CoA, and succinyl-CoA, through the action of alpha ketoglutarate dehydrogenase complex, through succinyl-CoA by the use of 1 GDP, production of GTP, we have succinate by the action of succinate synthase. Then fumarate is formed, action of succinate dehydrogenase. Then from fumarase enzyme के presence में malate is formed. Then malate dehydrogenase converts malate into oxaloacetate, which again goes into the cycle, combines with acetyl-CoA and then forms citrate. So this is the tricarboxylic acid cycle. Arsenic is one compound which inhibits pyruvate dehydrogenase. So blockages of how different presence of different elements affect. Excess of arsenic causes blackfoot disease. Excess of cadmium causes ouch disease or itai. Then excess of mercury causes Minamata disease. Excess of fluorine causes fluorosis. Excess of nitrate causes blue baby syndrome, or it is also known as methemoglobinemia. So the fate of 2-NADH in cytosol, it can either go into the aspartate malate shuttle or glycerol 3-phosphate shuttle. So when 2-NADH plus H positive gives rise to 5 ATP and 2-FADH gives rise to 3 ATP. Aspartate malate shuttle occurs in liver, heart, and kidney. Glycerol 3-phosphate shuttle occurs in brain, skeletal, muscle tissues. Regulation of glycolysis, basically what helps in positive regulation or negative regulation of glycolysis. This is the structure. So in case of glucose, conversion of to fructose and 6-phosphate and fructose 1,6-bisphosphate. These are the redetermining steps. Phosphofructokinase and back conversion of fructose 1,6-bisphosphate to phosphose 6-phosphate. These are the things which negatively regulate or positively regulate this conversion, and conversion of fructose 6-phosphate. These are the ones which positively convert or negatively convert to different steps. Then the next rate determining step is phosphoenol pyruvate. The conversion of phosphoenol pyruvate to pyruvate using the enzyme pyruvate kinase. Fructose 1,6-bisphosphate positively regulates ATP and alanine negatively regulates. And similarly, the conversion of pyruvate to oxaloacetate, action of enzyme pyruvate carboxylate positively regulated by acetyl-CoA, negatively regulated by ADP. Conversion of oxaloacetate to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase is negatively regulated by ADP. Pyruvate has one another. In case alanine is present, alanine inhibits the action of pyruvate kinase. So pyruvate is not produced. Alpha-1 antitrypsin, it is given for treatment of emphysema, swelling or anything. Talking about electron transport chain, it occurs in inner mitochondrial matrix. There are 5 complexes involved. One is NADH-Q reductase, or we also know it is also called NADH dehydrogenase complex. Second is succinate Q reductase. Third is Q is basically ubiquinone. QH2 cytochrome C reductase. Cytochrome B, FES, and cytochrome C1 are involved. Cytochrome C oxidase, cytochrome A, A3, and copper centers. Then complex 5, which is basically F-type ATPase. It is the ATP synthase. It has F0 and F1 complex. F0 is basically involved in oligomycin inhibition, and F1 is the first factor isolated from mitochondria. And oligomycin does not affect ETC but inhibits the ATP synthesis. Oxidative phosphorylation is basically the synthesis of energy-rich ATP molecule with the help of energy liberated during oxidation of reduced coenzymes, which are basically NADH and FADH provided in respiration. If we talk about electron transport, inhibitors of electron transport chain, if electron transport chain is inhibited, oxygen will not be consumed. So what are the inhibitors? Rotenone, amytal, pyricidin, antimycin, British anti-leucite, then cyanogen CN negative, carbon monoxide, H2S, sodium azide. So these are the steps where ETC is inhibited, and these are the inhibitors which inhibit ETC at the various steps. ATP synthesis is inhibited by these. First is orovertin. It binds to the F1 domain of ATP synthase. Then there is venturosidin. It binds to the F0 domain of ATP synthase. Then we have atractyloside. It is ATP-ADP translocase. One uncoupler is 2,4-dinitrophenol. It inhibits ATP synthesis by dissipating H-positive ion gradient without affecting the ETC. So it dissipates the proton gradient and it does not affect the ETC. Thermogenin is also an uncoupling protein. In presence of 2,4-DNP, oxygen will be consumed and ETC will not be affected. So this is it for the notes of carbohydrate. Hope it helped.